one thing that I like to talk about, so my favorite pathway in all of biology, this is the end, is called the pentose phosphate pathway. It's also called the hexomonophosphate shunt. And so the MCAT will use both of these, and it's really annoying. And they'll try to confuse you. So they'll even write the abbreviation. Like they'll write a question like, in the HMP shunt, and then they'll be referring to this pathway, and you'll be like, I don't know what that means. Well, that's this.
Yeah. Okay.
So the HMP shunt or PP or pentose phosphate pathway. And we'll talk about what both of those names refer to. So recall that glycolysis, we have a 6-carbon thing, 6-carbon and a phosphate, 6-carbon and a phosphate, and then 6-carbon, 2 phosphates, and then 2 3-carbon things, right? So I'd like to go over this again at the end just to drill it in one more time. So this is glycolysis, right? It's not that complicated, honestly.
No.
So once we go through once phosphofructokinase does its thing and we get fructose 1,6-bisphosphate, we're committed. But before that, we get to use these in other pathways, and this is one of those other pathways.
Okay.
So we take this, so this glucose 6-phosphate, we already know this can go into glycogen, for example. We just did that. It can go and become glycogen. It can also go in this other pathway, this pentose phosphate pathway or hexomonophosphate shunt. Now it's called a shunt because it's sort of coming off to the side. See, so like this is like, so that's what the shunt is. And the hexomonophosphate, well, we'll get to that again. So the key thing about this pathway is the enzyme.
Okay.
So here, the enzyme that does this is called glucose 6-phosphate dehydrogenase. Also called G6PD. So it's on the X chromosome, and it's the most commonly mutated gene in humans. It's big. It's a big gene, so there's a lot of potential places where it can be mutated, and it's the most commonly mutated gene. What it does is, so the products that it makes here are not important. What's important is it creates an enzyme, or it creates an electron carrier. And that electron carrier is NADPH. And both of these reactions, NADPH. This NADPH is essential for free radical reduction. Remember we talked about reactive oxygen species creating free radicals? So these NADPH can reduce those. They make something called glutathione, if you ever heard of that. So these make glutathione, which is then the sort of quintessential antioxidant in the body, right? So this is the quintessential antioxidant precursor. Okay, then, just for completeness, so then this makes something called ribose 5 phosphate. So that's why it's the pentose phosphate pathway, or so this is the pentose, and this is the hexomonophosphate, this glucose 6 phosphate is the hexomonophosphate, and this ribose 5 phosphate is the pentose phosphate, in the name. And then from this, you can create nucleotides, pyrimidines and purines. And remember, we talked about pyrimidines have a different synthetic pathway than purines. And that matters in terms of chemotherapeutic drugs, right? But they start from this precursor, ribose 5 phosphate. Or, there's some other things it can do, and then it can feed back into this. Okay, so let's talk about the mutation to this enzyme. So so this enzyme, even though it's big, as I said, the gene is big, and there's a lot of potential for mutations, we find that the rate of mutation in humans is higher than would be expected by random chance. When that's the case, when an enzyme is mutated higher than you would expect by random chance, what's going on there?
Wait, sorry, can you repeat that? Like that, just like your question.
Yeah, when you look at a population and you see an enzyme mutation, or you see a mutation, a genetic mutation within that population that's higher than would be expected by random chance,
Yeah.
what are you usually thinking?
Like adapted to it?
Yeah, right.
Yeah.
So there's some advantage.
Yeah.
There's some sort of, in some way, having that mutation gives this population an advantage.
Yeah.
And that's the case here. So having what's called G6PD deficiency, having G6PD deficiency protects you from malaria. Malaria is a parasite that lives in red blood cells. And red blood cells, remember, they don't have a nucleus, they don't have mitochondria. So the only metabolic pathway happening in red blood cells is this, glycolysis. And the only antioxidants being created are this NADPH and glutathione. Now, red blood cells are carrying a lot of oxygen, so they need antioxidants.
Yeah.
So they need this thing to function. When it doesn't, when there's a deficiency, the lifespan of red blood cells is shorter. So the average lifespan of a healthy, mature red blood cell is 120 days. If you have G6PD, the healthy lifespan may be 30 days. Now that gives you some disadvantage, but if you're from a region where malaria is endemic, then that gives you a huge advantage because the malaria can't live in your red blood cells. So this is more protective than the sickle cell trait.
Okay.
So sickle cell trait only protects against some kinds of malaria. This protects against all kinds. So this is way more common actually than the sickle cell trait. The thing about the sickle cell trait is it's more talked about because it's a very specific enzymatic mutation, or it's a very specific amino acid mutation. It's literally one base in the genetic code that's changed, and it creates this. Here, there's many different mutations. There's populations from Greece to Africa to Asia, Southern Europe, whatever, the Mediterranean, the whole Mediterranean region, Israel, Syria, whatever. wherever there's malaria, they have mutations, but they're all over the place within the gene. And then if someone has this, they take certain drugs, certain cancer drugs, sulfa drugs. What will happen is they'll get what's called a hemolytic crisis. So those drugs can be oxidizing, and all the red blood cells in the body will lyse. And so the key symptom is black urine, right? Black or brown urine, because all that hemoglobin now is filtering through the kidneys. It can really destroy your kidneys, let alone, you obviously have a severe anemia if that happens.
Yeah.
So that's bad. And so people who have this usually know it because that's happened to them a couple times. Actually, I've had students who've had this. And one thing that's interesting is you can't eat fava beans.
Okay.
You can't eat fava beans. So I always remember that for some reason. But there's other things that will trigger an attack also. So then the key thing is to remember this is on the X chromosome. So who's gonna be affected?
Women. Right? No, male.
Men.
Yeah.
Right? Because women have 2.
Women have 2 of the X, yeah, right?
Men just have 1. So if their one copy is mutated, that's it for them.
And that's it.
Right? And so cool. So this is this is the hexamonophosphate shunt. It's glucose 6-phosphate dehydrogenase, creates NADPH. This is a key antioxidant. The rest of the pathway, you can create ribose 5-phosphate to create nucleotides or feed back into here. And then it's the most commonly mutated enzyme on the X chromosome. It affects men, particularly in the Mediterranean region where malaria is endemic. And that results in red blood cells that are particularly sensitive to oxidative species and can undergo a hemolytic crisis with certain drugs and foods.